Okay, so everyone has already submit your homework 1. Excellent, everybody got 10 points. And then we will have another homework in the next two weeks. Okay, so same thing, you take a video, let me know, okay, you know those. As you may see already, this second homework has 2 parts. One is to explain the dot product, the applications, and your understanding of dot product between 2 vectors. Then I will ask you to explain how PCA we will discuss PCA next week. So after that, you just repeat what I taught about and explain PCA using your own language. Returned. So this is homework 1. I'd like to see most of you have a happy face here to reflect, okay, is this a goal? This is good. Let me know if you have any questions regarding the process of the lecturing and also homework. Also the computer session, whether we move too fast, you want to slow down, or you want to touch base on something else, you feel I'm ready to use some of those in my real work. I want to quickly cover some aspects that we can jump, say, okay. Just let me know. Any feedback, reflection will be welcome, appreciated. So today, I have agenda here. So that's in the channel this week, lecture 4. We are going to do some random number generator to do a simulation of the single-cell RNA-seq data. If you were here last week in the computer session, we tried to simulate a single-cell RNA-seq expression matrix, but we pump up those numbers. Just put 0, 1, 48, something like that from our mind. But today we try to use more theoretical way to, based on our understanding of the distribution of the single-cell RNA-seq data matrix. So we will make some more realistic simulation of the matrix. We will put that into a function, so you can write a small function for MATLAB, so you can reuse the code you have been writing to simulate the data. So next time you don't have to, from scratch, you have the function, and you just tell the function simulate how many genes, how many cells, a random matrix will be generated and look like a real one. Okay. Then we will visit the PowerPoint slides and then to see those metrics in terms of mean and variance, centrality. How can we do it in the MATLAB environment? And then very important, how can we compute covariance and the covariance matrix? So this will lay out the foundation for PCA. So PCA will rely on the covariance matrix between variables. And I will show you how we revisit this page. Okay, so from the previous slide. And then if we still have time, we will do differential expression analysis. See how can we download two data sets and compare them. Of course, these two data sets has to be comparable. It's either from the same cellular system, so they have shared cell type. Then you compare, for example, between wild type against knockout sample. So how can we do that? Okay, so you may know this before. In MATLAB, there are built-in function, RAND. So this is a function to generate a random number. So this random number is between 0 and 1 and uniformly distributed. Okay, so if you do this, you will be surprised on your screen. Maybe you generate the same number as I generated here. That's because this is a pseudo random number. It's not real. It's simulated, basically. It was algorithm. And our machine was initialized at the same configuration, so we generate the same number here. So it's not random. Okay. So if you see, okay, I'm doing the random testing. I did today, and I come back, turn my computer on, do again. You are not doing the real random simulation because you generate a series of random number which just repeat itself, just like a huge circle, and you have start from the position 1, 2, and you circle back after several rounds. So if I do that again, I generate another random number. So remember, those are the uniformly distributed number from 0 to 1. Okay. So if you want to generate 10 random numbers, okay, you just call once of this function, you will have this. And this function does not require any parameters to set up the distribution because distribution is fixed. The distribution is a PDF, probability distribution function, which is just like this. Okay, so this is 0, this is 1, and you have a frequency which is the same for every potential point in this range. So this CDF does not require any parameters. There's no shape, no peak. Okay, but if you want to have a normal distributed random number, you need to tell and where the peak is. So you have to give two numbers in order to describe this shape of the PDF. So for this case, we don't need any parameters, uniformly distributed. So now I'm going to change this to random n. Okay, random n will generate normal distributed random number with the mean in the middle. So the average will be 0, and the standard deviation will be 1. So you have you don't need any parameters because this is so-called standardized, okay, centered. Also, the range or variation has been fixed. So also, although this normal distribution is a parameter, require two parameters, but our function by default just use 0 and 1, so we don't need any parameters. So you can do the same thing. If you do that again, you get some negative value, Okay? The average will still around 0. You can do the same. See, I want to generate a 10 by 1, a list. Or you want to do 1 by 10, it's also work. Okay? Questions? Well, okay, right? So those are the continuous variables. So if you have this x, you generate y is uniform distributed numbers. So if you have this, let's see file. So we can see. If you want to see, okay, I want to use this as a single cell RNA-seq data random number generator. Can I use this? It's not very good because single cell data is a count. So we want to 0, 1, 2, 3, those integers. But here we don't. So this is not the best function or random number generator to simulate single cell RNA-seq data. You can do is if you see, okay, if this greater than 0.5, I will set logic. So I convert x into a logical vector. So this will help you to get a binarized 0, 1s. So any values in your simulator which greater than 0.5 will becomes true, it's 1. Otherwise will be 0. So there's some way you can binarize this. You set up a cutoff and then make this become a random single cell RNA-seq like data, but it is only 0, 1s. Okay. So you can set cutoff not only 0.5, you can set 0.3. Then in that case, you will have more 1s on average. Okay. So I want to first let's generate a lot of these numbers. See, 500. So now my x contains 500 uniformly distributed. Then you can visualize the distribution. Okay, so maybe we put 5,000, give a large number to make this histogram look better, looks more like a PDF. So what I'm doing here is to simulate, let the machine to generate 5,000 random numbers, and then I put this histogram bins. So those numbers have the range from 0 to 1. And I put them into 30 different bins. And if you have numbers located in this bin, the height will increase by 1. So this gives you a shape, see, where and how those numbers are distributed in this range. Okay, question? Which one? Oh, if you put a normal distribution, if you put an n here, you will get different shape because that follow the normal distribution. We will try that immediately, okay? So I gave another name. I want to see not only let's do y. So y, I use normal distribution. So these two functions look same, but because of the n is here, so the function you call will generate random number follow normal distribution. And it will pop up a lot of numbers which close to 0. Okay, centered around 0, they are negative values as well. So we can plot this histogram again and compare with uniformly distributed. So if you do that, you can see y is here, and bring this back, and you see this is different from the uniform distribution. Okay. Are we okay with those? Okay. Now let's just visit this negative binomial distribution. This is what we described. This is good for simulated single-cell RNA-seq data because this is for counts. This distribution is used for count. The function we are going to use is called this n bin RND. Okay. So let's see. This time we still do the x, but we are going to generate n binary RND. You cannot do this by itself because we really need so the spelling should be RND. Okay. You cannot do this because we need 2 input values to define the shape of this distribution. We can just follow the example shape here. So if you look at this, we need, if we have a distribution negative binomial 7 and 0.3, so 7 is the mean and 0.3 is the variance, then we need these 2 values, 7, 0.3, to make this work. Okay? So this time, there's a single number come out from this random number simulator. The first thing you will notice is an integer. So because this function is designed to generate counts, okay? So this integer. It's greater than 0, or could be 1, could be 0. It's a low probability because you define the average relatively high. If you do that again, you get another number. Okay? I can just repeat this many times. If you don't want to do manually repeating, you can see, okay, I want to generate 100 of them so this has become a 100 independent simulation, follow non-negative binomial distribution with parameter 7 and 0.3. Okay? So this will be, you can use this to simulate a gene expression. So how this gene expression level is, it will be, on average, will be 7, this gene expression. So if you look at this, this become a count, a list of count. And you see, okay, I simulate 100 cells. For this particular gene, I have an expression level 7. Okay? We can do, let's do 500 or more. See, 800. So I simulate 800 cell expression of a gene Now, if I do histogram for X, this shape is the blue, light blue one, okay? In the oh, by the way, the mean is not the parameter PRs. So this is this value is not the mean. Okay, so this is how many successful trials you obtained at the end. So you have to look at the definition of this, and 0.3 gave you the probability of success, and 7 means you obtained 7 success events, and then the number generated will be how many trials you need to achieve 7 success if you have a success rate of 0.3. So the mean you have to compute with this equation. That's theoretical solution. Okay, so what we can do is so this is the first distribution. If you have a second gene, say expression level is 10, let's do this 10 and 0.6. So you can simulate the Y gene, okay? 10 and 0.6. And 0.6. Normally we will fix this, put the 0.3 as the same, but here as an example, I just show using different p-value here. Then you can try Z. Z, if we use example here, you have 1 as the first parameter, and then 0.2 as the second. So I will do 1 and 0.2. Okay. So these 3 lines of code, X, Y, Z, and create 3 vectors. Each contains 800 integers, those counts. And they have different distribution defined by the negative binomial random number generator using these combinations of 2 parameters. So I can plot a histogram, put a hold on, and then plot the second Y, and then plot Z. So this 3 lines of code allow me to put all 3 distributions in the same figure, because after plot the first one, histogram, I hold on this figure and then allow the second one to be plotted and also superimposed onto the first one. Then I add 3, the third one. So hold on is a small command you can use to plot multiple figures on the same background. Okay, any questions so far? It's okay? Good. Now I'm going to write a small script. So script is a combination of several lines of code and put in the file. So next time you can run the script file instead of type that in again and again. So I will combine what we have tried and to simulate 3 genes expression in 1,000 cells to make a 3 by 1,000 matrix as my simulator. Okay. So what you need to do, because everything you write onto a command line here into this window, once you shut down your machine, it will still in the history of the command, but not runnable. So we are going to edit to set up my first script. So this, my script, you can give any name, valid name, to save the file. So this will create a file in this folder. So you see this has been created. And this editable space here will be the same as your command window, but it will save after you save this file. So your comments will be saved. So I will keep using the one which we generated here. So I'm going to do the first one. But this time I will change this to 0.01. And also I said 1,000 cell we want to simulate. So this is the first gene. And then I can simulate my second gene expression using Y. And then in this case, this gene expression level is higher than the first one. And I have Z gene. And this time I have a lower expression. Then I can do my histogram plot, figure histo. My X, hold on, and Y and Z. Okay, in this way, I have my first script in MATLAB had been written, and then I can clean my clear my variable space and close everything. Now, once you have this, it has been saved in here, and then you can see, okay, I want to run this. So you can click this button here. The active script file will be executed. And then you should have the histogram out, okay, like this. and 3 genes expression simulation in 1,000 cells. Then if you want to generate a whole matrix, you can put X, Y, Z into a big X matrix. In this case, you will have this big X. So we need to put them into, let's see. So this by rows, X. They're column by columns. So what we need to do is I can transpose all of them to make them horizontal, and then put them into the 3 rows. So this is our simulation. Okay, I feel this is not real, and because the real data has a lot of zeros in there. So I want to make some changes. Maybe I should put this into 0.1. Let's see. Even that, we shouldn't have zeros in there. Yeah, how can we make a lot of zeros in? Let's see, this time the number becomes smaller. So if my success rate is higher, then I have this. I think what we can do is just -1 from each of these to make this look more real. Oh, not good, because there are -1 come out. We don't want -1. So can I do this? Maybe make this even bigger, 0.33. Then this time, we start to show some zeros in the lowly expressed genes. Okay, so to me this looks more real. So if you consider this script, there's a limitation for this, because each time we have to manually put a 7, 10, 1, and we can only simulate 3 genes expression. If you want to simulate, say, 500 genes, then we need some more easier ways to do that, right? So we want to make this average expression, this parameter, this is not average again. This is in terms of negative binomial, this is the number of success events given the success rate 0.03, the number of trials. So so we want to make this into a variable. We want to use another random number generator to generate this value, to generate 7, 10, 1, 11, 10, 7, whatever, 1, 1, 1 for us. Okay? We don't want to type this every time. So we need to make this number, a parameter, become another number random number generator to give the value here. Okay, so this is the design of the simulator. So before that, let's make this into a function. So if you save my script, save as function. So you just change the name of the file. Then for a function, we need to define the keyword function. And if you type this correctly, the editor will recognize this keyword and makes it blue. So you are doing the right thing here, write a function. For the function, we need output. We need x as output. Then we need the same name. So you see the function and my script, these two are not very different. The only different is you put this line to indicate the code you are writing next is the function. So function become isolated, independent running unit. It will push to a special area in the memory to execute, and the only thing it will come back, give to you is the big X. Although you define a lot of variables inside, you even plot something out, but this function will call it. So if you run my script, everything will be show up in the workspace. But now if you do clear O, if you run this function, you give X out. You still have this figure, and the output will be just big X. So everything, the intermediate steps, all those variables have been encapsulated inside the function. Okay, so you put the function as something you don't care about the details in there, although you implement using your script a lot, but now you only care the output. So this function will generate big X matrix contains 3 genes and 1,000 cells. Is that clear? Maybe not, but gradually, if you haven't done any coding, you will spend some time in this for you to understand. But in general, function is a series of code, series line of code to execute and generate something and give you the output. The output is something you want to use for next step or the purpose of your coding. So if I see I need R3, so So function allow you to define the input. So here I ask 3 input. So these 3 input, I will use them to replace the R1, R2, R3. So this gives you some flexibility to define the function, let the function to generate 3 genes with given parameters. So the first gene, it was 7, 10, and 1. So this will be the same as you hard-coded when replace R1, R2, R3 with these numbers. You will see the same shape or you see before. But those numbers become variable. So if you do this, so I want to generate 3 genes with 2, 3, 4. So this time the shape of those values have been changed. So the number you pop up will be have different distributions, depends on your parameter you gave to the function. So that's why you need to do the function, because the function allows you to define the input variables and execute the code and replace those input variables to the intermediate variable values and then generate the output which determined by your input values. Yeah. Let's see. Yeah, function, I think this will be difficult because function itself is a variable. So if you put something else, maybe function A, will be better.
That's probably why it's not working.
Yeah. And then this one is still the function, still not my function.
Oh.
That's right. Yeah. Yeah. Then the formula you have to give the equals my function. That should work. Yeah.
Okay.
Yeah.
Or
Yeah. That should work. That's right.
okay.
Yeah.
Thank you.
Mm-hmm. And then because here you haven't defined the big X, so you need to define the big X, because big X is, I'll show you. So the big X should be, yeah, to put 3 lines together, 3 rows. Yeah.
Thank you.
Mm-hmm. So this is a toy function. We just want to use this to learn how to write a function, how to change the input variables, and get the output variables. You can comment this out because you don't want the function to generate figures all the time. You can use this as a debug, a diagnosis step, and to plot those out. But if the purpose is not to visualize the distributions, you can add those $sign in front of the lines to comment this out. So those become just annotation. There's nothing the code will execute for. So this way, you complete a function writing. Develop your own function to simulate 1,000 cells with 3 genes. 3 genes has a different levels of expression level. you can make this 1000 another variable. So I want to make n, so next time I can replace how many cells I want to generate. Okay? In this case, if you want to call this function, you need to give the last 200 cells. This will generate a 200. Okay? So 3 to 4 defines the 3 genes expression level. The larger number give you on average the higher expression of that gene. And the last parameter n here, because it was 1000 hard-coded, but now it becomes a variable, so you can play, even if you want to only generate 20,000, it's just the same, okay? It's just the same. All right, any questions? We finished this part. Write a function and change the parameters, customize gene expression level. Yeah, so this is very close to real application already. So if you know. Cause error, so what's the problem? Yes, looks like this my function. You should rename it. Yeah, so this if you fix it automatically, yeah, fix. That's right. Yeah, it doesn't matter. So because your file is called my script, this has to be the same as this function name. Yeah, yeah. So you are fine. And so just use.
So if I do edit function, and then I put this in my function.
Yeah.
Okay.
Yeah, you need to be the same name. This will give you the right one. Yeah, it's good. It's working, right? The reason you didn't work because you need to give the values. You need to input. You have to give these 3 numbers and 4 numbers in order to run it. So you see, when I call this function, I give 2, 3, 4 before you can run.
So I'll just say that I don't run it here.
Yeah, you have to run from the outside. Yeah. It's working perfect. Yeah. Okay. Okay.
So I basically covered everything.
Yeah.
Here is line 2, column 1, and I was trying to troubleshoot, but I'm getting the same error.
Mm-hmm. Yeah, if you look at the source code here, you have, so that's within your file, my function files. And then you don't need this extra lines. Okay, now it's fixed.
Okay.
You don't need this, so you can put a percentage sign in front of it.
Make it as a comment?
Yeah, comment out. Okay, so that's then you finished here. So you can you can delete the rest. Okay, then you save. So that's your definition.
My function.
Yeah.
Okay.
So this name has to be the same as a file name.
Also like .n.
The same.
There's a .n at the end.
No dot. So this is okay.
Okay.
Working. And then from here, you need 3 numbers as input. So this should work. Working. Yeah. Yeah. If you change this to different numbers, then you generate different numbers of cells. Okay. It's working. Okay. Yeah. Good. Okay. Yeah. Not there. Not yet. Slowly. Okay, Sarah. Okay. All right. Let's see. So now I'm going to show you the function inside the toolbox which doing this simulation. Very similar to this, but I want you to look the real implementation of this simulation. So this function is called SC sim data, okay, simulated data. This allows you to simulate X. So I'm calling the function, so I know how the function is writing. If you wait some time, allow this system to show you the usage. So you will see, okay, the first number I will give how many genes I want to simulate, and the second will be how many cells I want to simulate. Okay, and there are different type of the method to do the simulation, but I only need, say, I want to simulate 100 gene and with 200 cells. So this should work because the function has been developed, and then you can use. Okay. So if you look at the output X, it contains a matrix looks like this. There are some genes. The second gene, for example, in my simulation have a higher expression, and my 6 gene, number 6, number 7 have a relatively low, a lot of zeros. So this is a way that we can simulate whatever number of genes cells, and look like there are some expression level variability across genes. Okay, so that's something we want to try. In order to see the source code of this function, you have to tap edit. Remember to tap edit. But if this function has been already written, you will not pop up an empty file. But this will show you the source code of already developed function. That's within this function, you see the definition of number of genes, number of cells as a variable, input variable, and the methods. So the method is just indicator and which method we are going to use to do the simulation. If you don't type anything by default, this will be the simple and the very simple model. Let's go move down, and then first this output variable X will be given a matrix, they all zeros. Okay, that I just predefine the empty space for this variable X. And then I start to decide what type of the method we are using. If the case is a simple method, then I will do the following. I will have a shape parameter and a scale parameter defined as 2, 1/2. Then there's a gamma distribution random number generator. So the shape and the scale and number of genes. This will give you a list of the random number of same as the number of genes, and then this random number will be using to determine the average expression of those genes. So sometimes maybe 22, maybe 3, maybe 1, 0. So this gave you the average. So for each of this average, I will use, so this will be used in, you see this already. So sometimes this will be 7, sometimes 10, sometimes 1, determined by this random generated number. And the p is fixed. I fixed it into 0.1. So this is exactly you are doing, you have been doing to simulate the data, and then simulate a row, okay? It depends on how many cells you want to simulate. So I just do a loop and do for all the genes from 1 to the number you want to simulate it, and then add it row by row, and that's all. Okay? So only different from what you are writing, you simulate 3 genes. So you define these 3 genes as a expression level 2, 3, 4. So in my case, rather than put 2, 3, 4 manually or something else numbers, I generated this 2, 3, 4 numbers from gamma distribution to give me some variability of the gene difference expression. That's all. Okay, very, very simple, right? Okay. So that's also the function we used in the toolbox if you have this on. Now in the input data, you can do a simulation. So here is the function. That function will be called. So you are not using any real data or download or provided or yourself example. You just simulate the data. So in this case, I will simulate 3,000 genes, 5,000 cells. That's only number we need. And then very quickly you get a matrix. And this matrix will contain a lot of genes. So this is simulated data. If you look at the name of genes, it's just by numbers. Okay? Because we simulated, we don't have a name for those genes. They are just random number vectors. Okay, 5 minutes break. Okay, I hope you get some idea how to develop or write a function. So MATLAB requires your function name has to be the same as the file name. So this file is called sc_simul_data. So your function is also called sc_simul_data. It does not allow you to do something like this. Okay, the dash will be minus. Okay, so you cannot use operators in your function name. It has to be valid. So underscore is a safe variable name. Character to be in the function name. Yeah. So let's move to the next task in our schedule. So I believe it will be descriptive matrix about variables. So let's just use let's simulate x as my input to do those. So switch off this. Okay. Yeah, just simulate 100 genes with 200 cells. So we know you can compute a mean and see, okay, you have an average of, so use the function mean. This will be by the rows. Okay, this will be the genes average. So let's see mu. So if you get the values, you see this is by 100. Okay, 100. This by rows. If you do so, this will be the library size if you do the sum of x by columns. Okay, by definition is how many rays appeared in one cell. So by cells are in the columns. So we just sum the total. That's the library size, basically. So in our case, we have 200 cells. So the library size looks like this. So what I can see, the highest is 832, and the lowest in this view, they're 620. So looks like we need some normalization for this data, right? So to do the normalization, we will divide each column to its library size. So let's do x norm. So x divided by library size. Now, if you look at this xn, which is a so-called normalized version of, you see this become fractions because they've been normalized. Okay. And now, if you do x n So in this case, you compute the library size for the normalized matrix, then you will get the answer will be all ones, okay? Because you already divide by the total. But this number is too small. Normally, you will multiplicate this by 10k. So you compute the total for each column, then divide elements in each column by the total and make them normalized. But normalized, because the fraction is too small, we multiply it by a factor 10,000. Now this becomes your expression level normalized version. Okay, so xn. Now the summation of each column now becomes 10,000. Okay. People also like to do a log transformation 1p xn. One p means every value plus 1, and before you take a nature log. To guarantee, because nature log 0 does not give you anything, so this plus 1 just to make you to plus 1 for any values to give in there, so this is still 0. So that's a function 1p means. Okay, so what I just showed you is to do this normalization and then to do a log 1p. So this is the whole thing is normalized log transformed. So this is the normalization method and the transformation normally you should do. And it's not too much and not overdressed this problem. There are many different other ways to do normalization, but many of those methods tend to give you over processed results. So you want to keep everything simple. So normally this will be satisfied enough to give you reasonable normalized data for downstream most of. So for the gene expression, we know you can calculate the mean. And for the variance, you do variance and also give some of this. So this will compute the variance of the gene expression before transformation. Okay, remember X is the raw UMI count we simulate. They're not transformed. So you can do a normalized version of this, put a xn in there. Okay, you can do the view with xn instead of the raw data. That's very basic. Okay, you do you compute the mean for the UMI before normalization, or you can do after you did that. Library size normalization. And this number 2 here just indicate I want to do by rows. If you don't put that, by default it's 1, and everything is by columns. So in our case, we want to compute the average expression per gene. We don't want to compute average expression of all the genes in one cell. Or depends on your problems. You may want to compute the average of those by cell. Variation, okay? So what is the standard deviation? SD, it's very similar to variation SD as STD, same format. So once you have SD, you know the CV, the definition of CV is so remember, CV is a standard deviation divided by the mean. So once you have your mean is here, your standard deviation is here, so you can compute the CV, use SD divided by the mean. Okay, so this is a commonly used measurements for the variation. So variance, standard deviation, or CV, coefficient of variation. So you can use any of them to do this. So for these 200 genes, I have an average and a CV. So these two vectors contain the same number of elements. What I can do is to do a scatter plot to see the relationship between CV and the mean. So this is a figure. You have a beautiful curve here. Okay. So in the single cell data, although we are simulating the results, and it seems there's a nice curve which defines the relationship between average and the CV. So they are negatively correlated. What that means is if you have a gene expression level is high, which you to the right side of x-axis, this highly expressed gene tend to have lower coefficient of variance. This curve is so clean that's not normally we will see from the real data. But this indicates there's some internal link between these two variables. That's why I gave you such a beautiful curve. There's no gene we simulate to jump away from the curve. So you don't see a gene here. They all follow this pattern nicely. So in real data, if you see something like this, you need immediately raise up your alert. It's, in the system, these two the variables are correlated. They're somehow linked. There's some underlying principle allows them to be linked, give you something like this. Otherwise, you shouldn't see this, okay? We can try real data and see whether these two variables are linked like this. Let's see where we can find a real data set. Remember, our data is simulated, follow some distribution. There's some noise because of the generation of the random number generator, but not much. Most of it follow the distribution we just described. But let's see a real data set. I will do inside here in the example data. I will click this to load. So if you move around in this current folder and point to the right example data, you can load this into, give you SCE. So now I'm using a real data which from SCEX. This from the NCBI geodatabase download. So now this X is a real data, okay? Contains 7,173 genes in 2,075 cells. So I'm going to do the same, what I just simulated for this, and look at the mu and the CV relationship. To do the normalization, and I use this function, okay. Yeah, I need a library size. Let me do that again, okay. So I load example data and read out the expression matrix, and then I need to define the library size for this and do normalization. Okay, then I define the mu, which is the genes expression average, and then define the CV to do SD again. Define the CV for the gene and scatter plot. Okay, so this is from the real data. Make sure you understand each point in this figure is a gene. Okay, so this gene, how many points in there? There are 7,137 points in this plot. And some those genes have a high expression. This level is high, okay? And some genes have low expression from this end. Some are almost close to zero, very small expression. So once you have the high expression level on average, then your dispersion level, which measured using coefficient of variance, is also small. Okay? And since there's a boundary here, there's no gene, even from the real very noisy expression, real data set, there's no gene will be able to jump inside this empty region. There's some limitation mathematically, not allow them to go that region. So you do see a curve which with a boundary here. I'm going to do a transformation in the log, and then you see a straight line, and some of the genes jump out from the cell line. So let's do the log transformation figure. Yeah, you receive so you transform the curve like that into a almost straight line. Okay, so this is almost. So if you look at this, there's some of this level of this bending down at the end when this expression level is high. There's theoretical solution for this problem is there's a straight line here. We are interested in genes which jump away from this theoretical curve. Those genes are interesting ones. Yeah.
What is
Oh, this is just indicate the shape. You want to use a circle to show this. If you put this as a S, you can replace, and then you will see the square. Or if you put a triangle, something like that, just define the shape. If you put a dot, then it will be filled up. Good question. Yeah, so just remember this figure and from real data. We are going to come back to this relationship later when we define the way to find the highly variable genes. So those genes are special ones, jump away from this regression because you have 7,000 genes. Most of them fit this curve nicely. And those special ones are functionally more important. They jump away from the curve, either for the same level of the average. Those genes have a higher than expectation variability. So those are the genes that should not have such a high variability, but they do from they jump away from the curve, which theoretically they shouldn't. So if you do the same thing for the simulated data, then there's no gene will jump away from the curve. Everything perfectly fit on this analytical solution. But that's something that we will touch base on later, not today. Any questions for this part? We need to move to compute covariance matrix, okay? Okay, so here is the equation, how you calculate the covariance if you have two variables. So variables, once you have a data matrix, you have samples, you have variables. Variables define the sample's properties, right? So the covariance matrix is between different variables, okay? In single cell data, if we consider cells as a sample, then the genes will be the variables define the property of cells. So genes will be the variable. So covariance normally in our language will be defined the relationship between genes. So one gene one, gene two, whether they correlated or covariant or not. So we use this to compute. So if you have a gene A, it's like a vector, and their expression level in many, many cells, then gene B, and we can compute their covariance between them. And this two has to be the same number, so you have to sample the same gene in the two genes in the same cell. Okay, you cannot do one sample against another sample. You have a different number of cells. They are not measured in the same cell. You cannot do that. So this is within the matrix of the same data set. Okay. If you put that two genes vector in there, this will give you a covariance matrix. This is a covariance between gene A to itself, gene B to itself. So it depends on the variability of those gene expression of the same gene in different cells. You will get either larger or small variance of the gene expression. Then you consider this off-diagonal position or the covariance of these two gene relationship. Well, this diagonal position are just variance of the same gene itself and the variability in the different cells. So I'm going to copy this. I just highlight and copy A and B and put in here. This is the covariance matrix for two variables. So if you do variance for A, so this number will be 2.33 in this corner here. And the variance B is this 123. So when you compute the variance A and B itself, you are not using any correlation between A and B, just by itself. So how variable is there? And then you measure the covariance that's between A and B. So this is the value. Now it's your job, okay? In MATLAB, you define X and Y, two variables. And let's compute the covariance between these two. just make sure you convince yourself and you can get this covariance matrix using the function, or you know how to do this step by step yourself. If you know how to compute the mean, actually, right? You have a mean of x, and you can do x minus mean. So you can use two different methods. One is just use built-in covariance function, COV, to compute xy's covariance. Or you can do this step by step using the coding skills you just learned. And to do this, once you do this centralization, okay, you centralize your data around the mean, then you get two vectors. These two vectors, you can do the multiplication and summation, and then divide by. You should get this number. So if you know how to do it, give it a try. In MATLAB. Yep. Okay, I saw most of you know how to use this function directly to obtain the covariance matrix, which the values at the corner of the position. So let me very quickly type in xy. Okay, so x, y contain the same number of values. Is that correct? Yeah. So so now I normalize, centralize x by minus is and then so this is my centralized. So when I do this, I basically obtain this third column here. And then I will multiplicate this with y centralized. So this has to be element based. So otherwise, I'm multiplicated with two vectors. This will give me error because the inner dimension has to be matched. So maybe I should do that. But in this case, I just do element based. Okay. Let me do the summation of this and then divide by n minus 1. So how many? 9 minus 1. So this should be giving the results. So the formula is exactly the same here I described. Okay. Of course, you can use built-in function without explicitly compute all these different steps. And then x, y should be, so this function gave you both the variance of x itself, variance of y, and also the covariance matrix. All right. Remember, you have to centralize your data. Okay, doing this step is very important in computing covariance matrix. I'm going to try whether I can use this matrix multiplication to do this. If I do this, I will transpose the second one. This gave me the dot product. And then I divide by nine minus 1. I don't know if I can get the same. So I hope you understand this and this. You get the same value, the covariance matrix, but here we use the dot product. Okay. I don't know which formula you like most, but this is the better way to do dot product rather than element-based summation. The results are the same. Okay. So, what's the covariance matrix you can compute for using dot product? You just do. You have A dot B, and it divided by the n minus 1. So this is the covariance matrix. Oh, A and B has to be centralized. Remember, you cannot use X times B transposed after you centralize this. Okay, so let's put a bar here. So this is the covariance. Also, you have to remember A and B are the genes. Okay, they're variables. And then sometimes you do by cells, that's by sample. You can also compute this. In that case, the dot product will be the two samples cosine distance, basically. Okay, they're different meaning. Okay, so this is for the variables, the relationship between genes. If you replace with the samples, that will be the distance between two cells, dot product of the two cells. When you do dot product between two cells, you don't need to centralize them. You can just use the raw data and to get the distance. Why you can get a distance? The reason is the formula we had last week. And that's also your homework. I'll show you what I'm talking about. Your homework part 1 is to explain the application of this. What I talk about here, if you play with the dot product between two cells, that's two samples. The dot product is this. It will give you basically cosine. Cosine can be used as a similarity measurement between two vectors. If you two these vectors are the similar lengths. So you can divide by this norm. So you pull them back to the unit circle, and then to get a cosine. So when you apply dot product to two samples, especially you normalize each sample by its norm, you get the similarity between these two samples. So, but this week we are talking about the variance between two variables. That's if this is considered as the two genes, then this one also normalized, centralized, then it becomes the covariance between two genes. There are some links between them. Yeah, the covariance you can also compute with the dot product here. So your homework, I want you to explain what that projection means. Okay, you project A onto the B, and using this equation to get A's coordinate, using B as your x-axis. Okay, so you very quickly compute how long from this point to here. If you have that concept on your mind, it's all, you are okay. Let's see, we move to next. I don't think we have time to do the DE analysis, but I can quickly show you using the toolbox what you can do. Okay. So I just start my interface here. I'm going to use example data to show you the differential expression analysis. So from here, we can, using the geo accession number. In this case, we put two numbers because we need two samples to compare with. So the good example will be the one which on the top here. So I just type that in to replace this. It would be 3308 5, 4, 7. And then I put a comma here and then 8. So you can search, and these are 2 different samples from one study, and they want to compare. So this program will go there to download the 2 samples data and merge into one and label the cell differently. There are many other ways you can pull or merge 2 different samples together, but this is the easy way. You don't have to touch anything, and automatically you get the separated 2 groups of cells from 2 different sources. Then we are going to annotate the cell type automatically, and then start to compare across these 2 samples for any given cell type to find the differentially expressed genes. So in the middle, you will have a chance to select which method you want to do the normalization. Whether you want to impute your data or not, although it's not encouraged to impute any data. But there are options. If you want to use imputation, you have the choice. Okay, it's done, and I will use automatic procedures to do all the downstream small data, I believe. So if you look at this feedback message, basically the procedure is doing under the interface. We do have to download and process those data files separately, and 2 samples. Then the library size normalization, you know how to do it, okay? And then log transformation has been done. So this is annotated cells, and then with different cell type from samples, and then you can label the cells with a batch ID. We have 2